Okay, we're going to jump into our speaker here, and John Icky will introduce. That would be you, John. Yes. You can either hold this like this, or I can pin it on you. Yeah, no, it's length doesn't matter.
Okay. Okay, in order to put this introduction into perspective.
Hold up the lavalier.
In order to put it into perspective, it takes us back nearly 80 years. Dick and I went to Highlands Mendota Beach, which is now Crestwood School. His sister Kitty was in my class, so it goes without saying that our history is a long one. Now in the present, I introduce him as a professor emeritus of veterinary pathology after retiring from the faculty in 2014. His specialty is ocular pathology. He's particularly fond of comparative ocular anatomy and an enthusiastic student of vertebrate evolution. It is this interest that has inspired today's talk. I thank him for coming and invite him to expand on vertebrate evolution. Dick, the stage is yours.
You can ignore it. Let's try that.
Okay, thank you very much. Welcome, everybody. Welcome those of you at home on the Zoom channel. And I hope you enjoy a little bit about evolution, particularly evolution of the eye. So as you can see from the picture here, the thrust of my talk is going to be to try and explain why, in terms of vision, why mammals, the default situation of mammals is to have a good vision in either dim light or nocturnal, and the default vision in birds is to have vision in the bright daylight. Of course, there are exceptions among both birds and mammals, but generally speaking, the mammalian vision is best functional under dim light circumstances. And nobody's taught me how to advance the.
Richard, just.
Oh, you advance. Next slide.
Okay. Come on. Just I'll get it.
There we go.
So just a little bit about what I do. I'm retired in 2014 from the pathology faculty at the veterinary school at the University of Wisconsin. And I started a lab called the Comparative Ocular Pathology Lab of Wisconsin, which is a mail-in eye pathology lab. So most of what we do is pathology of eyes. The pie chart on the screen right now illustrates the species which we see most commonly. Our clients are veterinary ophthalmologists from around the country, and the specimens we receive largely are specimens from the eye, most commonly globes, where there's been a medical decision to remove the eye. So far and away, our most common specimen would be a canine specimen, followed followed distantly by cats, followed again distantly by horses, the three most common domestic species that we deal with. And then there's another 5,345, which we just say is exotic, and that includes everything from fish to humans. The next slide, we'll just take that exotics, and now we're going to jump back. The previous slide, just a few days ago, I calculated the numbers. This slide, the numbers were calculated in 2016, so you'll see that the numbers are a little bit smaller. But this takes those animals we call exotics and makes again a pie chart illustrating the group of animals, the group of vertebrate eyes, most of them vertebrates, a few invertebrates, from which we have the specimens. And again, you can see that mammalians make up the bulk of our collection, followed distantly by birds, and distantly again by reptiles. And it's going to be mammals, birds, and reptiles that we're talking about mostly here. Next. And so when we receive an eye at the Copal lab, this is basically what we get. The example of the eye that we have in the middle is an eye from an elephant. And this is the type of material we'll receive. We'll receive a globe. We often photograph it so we can document the size and the size of the various components of the eye. And then on paperwork, we have the age, the sex, the gender, something a little bit about where it came from. And if there's a disease, we'll have something about the history of the disease. Most of what I'm going to talk about now is information I've gleaned from looking at normal eyes rather than diseased eyes. And then in order to study the anatomy, what we do is we process these eyes in such a way that we can look at them in a microscope slide, which is illustrated below. That the eye that's on the board here is an eye from a mongoose. And so we can look at on a microscopic level, we can look at the various characteristics and categorize the anatomic features of the eye. So that's the type of material that I have used to put together some of the thoughts that I'll present today. Next. So this is just a mammal eye and a bird eye. And I've chosen two species that have eyes that are adapted for their particular group. There are eyes that are adapted for maximum visual acuity. And that would be the primates among the mammals. And this happens to be a cynomolgus monkey. And the diurnal raptors among the birds. And this happens to be a red-tailed hawk. And you can see that there are, just looking at it, there are some differences. I'm going to categorize some of the major differences between mammal eyes and bird eyes, and virtually every feature that I'm going to mention is more sophisticated, more complicated, more functional in the bird eye compared to the mammalian eye. Again, both of these eyes are adapted for a high degree of visual acuity in daylight circumstances. So the primate is kind of the exception among mammals. It has eyes that are adapted to maximize visual acuity during the daytime. I think I'm going to just point out a few things, and hopefully I'll speak loudly. Hopefully people in the room can hear me. So in the bird eye, things that we'll pay a little bit more attention to in the future is, one is notice that the overall shape of the eye is different. In order for the eye to have a shape that's not spherical, there has to be a bone in the eye, and you can see that right there. There's a bone, and we'll cover that. The rest of the outside of the eye, in the mammalian eye, is a structural protein called collagen. In bird eyes. No. Please still clip. Thanks for the thought, though. So the bird, as you would see in virtually every type of vertebrate eye, other than the mammal, the portion of the eye we call the sclera is made of cartilage, whereas in the mammal it's just collagen. There's also a difference in the lens. You see a portion of the lens on we would call this the equator of the lens, that's specially adapted, and that is it's called the annular pad. It's an adaptation that makes focusing of the lens quicker and more precise. This thing that's fairly obvious here, I'm not going to talk about. This is where the blood vascular supply furnishes the inner portion of the retina. Next. So now we're going to delve into a little bit about evolution. I want to give a shout out to this website called the Time Tree of Life. It allows you to put together your own phylogenies. You just put, if you put in 2 species, it'll give you all the scientific data that and an estimation as to how long ago they shared a common ancestor. If you put together a whole group of species, it will print out a phylogeny like this. So on the right are the common names of all the various extant or still living vertebrates. And then on the bottom, you can see the time zone, to the timeline. These are in millions of years, so 400 million years, 200 million years ago, and the Cretaceous, the Jurassic, and the Triassic were the ages of the dinosaurs. And a couple things I want to point out. This notice this area here. This is where modern mammals evolved. Before that time, mammals in the fossil history were rare. So there's not a whole lot that's known about them. Notice on the top is the monotreme. These are the egg-laying mammals, and the marsupials and placental mammals split off from the egg-laying mammals in the early portion of the Jurassic. So when dinosaurs were still around. Now, all of the marsupials and placental mammals have eyes basically identical to the eyes that I showed you before. And then we're gonna concentrate a little bit on the duck-billed platypus eye because it has some interesting different features. Notice this long line here. These would be animals that are committed to the mammalian line, but very little is known about what their eyes were like because the eye doesn't preserve very well in the fossil record. But they split off from the group that would become reptiles and birds way back before the time of the dinosaurs, way back even before the Permian, and they split off from animals that would be perhaps categorized as amphibians today. So both of these groups, the reptiles and the mammals, acquired ocular characteristics that should have been present in animals that eventually went on to become amphibians. Of course, we don't know what they were like back in the time that they shared a common ancestor. Another thing to point out here is this group here: birds, crocodiles, and turtles. And this long line here, all this in here, every kind of a dinosaur that you can think of has split off in this line that eventually ends up with just the birds. But the crocodiles and turtles split off at a much earlier period in time. Unlike the lizards, I'm not going to talk about the tuatara. Unlike the lizards that split off way back in the Permian, well before there were any dinosaurs at all. So birds and lizards, iguanas, monitor lizards, geckos, chameleons are related, but way back in time. And I'm just going to say it now because I'm not going to talk about reptiles anymore. Many of the features, most of the features that I'm going to talk about that are sophisticated for vision in the bird are also shared by the lizards. So again, they've inherited their inheritance from evolution includes many of the features that make them have great eyes in daylight circumstances where they have plenty of light, and we'll talk about perhaps why that is. Next slide. So these are some features I'm going to talk about. In the yellow, these are traits that are seen in birds, reptiles, and amphibians, but are not seen in the placental mammals. And we're going to include the marsupials in that group as well. So cartilage in the sclera, we talked about. Photoreceptor oil droplets This is a feature that allows the cones of the retina to have advanced color vision, which is seen in all of these animals, but not the placental mammals. And then shading of the photoreceptors. This is a feature that allows the photoreceptors, the cells in the retina that sense light, to avoid glare. If they have too much light coming in the eye, it allows them to avoid glare. Again, amphibians, reptiles, and birds all have that. The placental mammals do not. In the blue are features that we only see in the reptiles and birds. Those would include, I showed you the bone in the sclera, the annular lens pad that allows for better focusing of the lens, and then skeletal muscle. The other animals all have smooth muscle, and there's great advantage to having skeletal muscle control the functions of the eye. And then in the green are a couple of features that are only seen among the placental or marsupial mammals. And we're not going to talk too much more about those, but those are features that mammals seem to have developed in the period of time when they separated from the reptiles. Next slide. Now let's fold in the egg-laying mammals, the duck-billed platypus. And recently we've also gotten echidna eyes, so I can say that most of these features apply to the echidna eye as well. So the duck-billed platypus now has cartilage in the sclera. It has oil droplets in the retina, and has shading of the photoreceptors, making it basically the equivalent of the vertebrates that inherited the traits from the amphibians, which, if you think about it, would fit with that evolutionary phylogeny that I showed you. It all goes back to when mammals, when vertebrates first came on land, they evolved from lungfish, and then they became something equivalent to an amphibian, and then split off into two groups. Eventually it became reptiles and birds. Another group became mammals. Next. So here, I'm just going to quickly show you the traits that I'm talking about. And this may or may not mean much to you, but cartilage in the sclera on the very top here is an image of what cartilage looks like in the sclera. We do not see cartilage in placental or marsupial mammals, but basically we see it in everything else, going way back to sharks. Everything else has cartilage in their sclera. Bone, this is a section of a bird eye. There's the bone, it's called a scleral ossicle. And then in addition, this shows this is a stain which makes the skeletal muscle stain red. And you can see the skeletal muscles in the bird eye. Much more functional for a number of reasons than the smooth muscle that mammals have. The equatorial portion of the lens has this specially differentiated structure which helps to focus the lens, called the annular pad. Photoreceptor oil droplets. This is a high resolution image of a raptor, of a hawk retina. The thick cell outer segments here are cones. This happens to be a double cone, but then at the edge of what we call the inner segment are these little structures here, which are oil droplets, not seen in placental mammals or marsupials, but seen in birds and reptiles. Features that we see in mammals, but not in others, is blood vessels in the retina. If you see actual blood vessels in the retina, then you're looking at a mammal, because no other vertebrates have that. And then many mammals have a reflective structure in the back of the eyes. It's why, remember the raccoon picture I showed you? It's why when you shine a light in their eye at nighttime, it reflects back. Not all mammals have that, and there are some other animals that are not mammals that have it, but it's generally speaking a characteristic of mammalian eyes. And then this picture doesn't show it because this thing is in the way, but There you go. You can see the dark pigmentation here is shading the outer, the portion of the retina that interacts with light, and that helps to cut down on glare. Seems like a pretty simple thing to evolve, but placental and marsupial mammals don't have that.
Can we ask questions during this?
Sure.
The mammalian blood supply, how do the reptiles get nutrients into the eye then, or is it a size thing?
It's partly a size thing. Remember that radiator-like structure I showed you in the bird eye?
Yeah.
That's how both birds and iguanas, lizards get structures. Some invertebrates, rather than having blood vessels actually in the retina, will have blood vessels right next to the retina, but not in the retina, but in the vitreous. Others will simply acquire their nutrients from the back of the retina.
Okay.
Next. So now we're going to fold in the duck-billed platypus, and like I said, we now have echidna eyes, and so these are some features that we will see in the duck-billed platypus eye. They now have double cones. They have oil droplets, and to be fair, we do see these in small numbers of cells in the marsupial eyes as well. We see shading of the outer segments, so we had lost in the rest of them, and now we see cartilage in the sclera again. So a lot like what you would have expected mammals to have inherited from their amphibian ancestors. So we go back to a phylogeny. Here is the split between animals that eventually would become mammals and animals that would eventually become birds and reptiles. And then here is the split, which happened fairly quickly in the middle of the era of the dinosaurs, when mammalian species split between marsupials and placental mammals. And then we have basically two types of egg-laying mammals that are still alive today, the duck-billed platypus and the echidna. And then we have this long period of time here when we know virtually nothing about what the eyes would be like. Interesting, though, that these animals maintain eyes as you would expect them to appear, as if they were inherited during this split back here. And again, along this line here, as all the dinosaur groups that are now missing. I wish this would have been a lizard instead of a snake, because snakes don't really fit very well in what I'm trying to explain, and I'll touch on that at the very end. Next. So this is a duck-billed platypus eye. What we're showing here is the cartilage in the eye. You can see it in this growth specimen as well. This is the lens. There's no nuclear bow. There's no skeletal muscle in the eye. So it has features which today would be like mammal or also like reptile. Next. But the reptiles have skeletal muscle. And then this picture doesn't show it very well, but if you're a student of the retina, you'll recognize that there are both double cones and oil droplets in that, and then you can pretty easily see that there's this feature of shading of the photoreceptors in the back of the eye. So the egg-laying mammals retain some of the features that they might have inherited from their amphibian ancestors. And I'm using the word amphibian. They probably weren't amphibians back in those days, but the common ancestor with both amphibians and modern animals. Next. So we're going to look at this again and point out a few features that are happening. One is these animals down here are cold-blooded. And what's the feature of that? If you're a cold-blooded animal, living during this period of time back in the Jurassic and Triassic era, then by virtue of your metabolism, you need to be in the bright sunlight, because in order to warm yourself to the point where your metabolism can function, you need to bask in the sun and allow the sun to warm your body. So if that's your living circumstances, and that's where you're going to evolve a visual system, you're probably going to evolve a visual system that takes advantage of plenty of ambient light, which concentrates then on an image forming eye that features high resolution visual acuity and not functional vision under dim light circumstances. Now, it's controversial, but there are a number of paleontological reasons to believe that even back in this era, the animals that were in the lineage that eventually would become mammals already were homeothermic, were already warm-blooded. And if you're thinking about surviving between these two groups, if you're warm-blooded, that allows for you to have access to nighttime survival. So probably already, well before the split that happened here, the group of animals that were destined to become mammals probably already had a bias towards being dominant at night as opposed to during the day, which might explain part of the reasons why these egg-laying mammals don't contain features that would suggest more sophisticated vision. Now, these animals, so nocturnal animals, by the time you get in the Jurassic, you have to think about predation. Yeah, because dinosaurs are dominant. Now, predatory dinosaurs are dominant. So first of all, there are not very many mammalian fossils in the early Jurassic through the Cretaceous. And second of all, you have to think about lifestyle adaptations that allow you to avoid dinosaur predation. And that's what they think has happened in this area here. Somehow these egg-laying mammals, the mammals that existed before, their existence became more and more threatened because of dinosaur predation. They become rarer and rarer, and eventually the only ones that survive, maybe even just a small number of species, are exquisitely nocturnally adapted or burrowing animals that live underground, and the use it or lose it principle in evolution comes into play in terms of the eyes. If you're existing in the dark and or underground, and you don't need eyes, you're probably going to lose ocular sophistication, and that's what people think has happened. I don't know what duck-billed platypuses were like back in this days, but if duck-billed platypuses have the same habitat or lifestyle that they have today, they come out at night, they live in burrows, and they spend their nighttime hunting with their eyes closed in small running streams. So with that as a lifestyle, they probably never saw a dinosaur. If they lived during this period of time, they probably never saw a dinosaur, and it makes sense that they would probably still be around. Are there any questions about that? That's kind of the meat of.
About 5 minutes.
Okay, next. So these are some paleontologic features that make people think that the early mammalians were warm-blooded, and they're kind of subtle. The pineal gland is like some people call it the third eye. It's used in animals in order to induce basking in the sunlight. So when that disappears, they think maybe animals don't have to bask. Body hair is lost in the fossil, but if you look at coproliths, petrified poop from dinosaurs, you can find hair in the coprolith coprolith. And the presence or absence of whiskers can be deduced by finding this big opening in the skull here where the trigeminal nerve comes out, and that's the nerve that feeds the whiskers. So if you got whiskers, you figure you got hair. And then I didn't really talk about lactation. Let's just skip that. Next. So this is the theorem that during the Cretaceous period, when the dinosaurs were dominant, mammalian eyes, mammals survived by either going underground or coming out only at night, and sophisticated ocular features useful for visual acuity vanished during what they call the nocturnal bottleneck as a survival mechanism to avoid dinosaur predation. And then when dinosaurs left, then mammals, 66 million years ago, then mammals radiated to what they are today, including a number of different niches. Next. So let's look at a mammal who has undergone this degeneration of their eyes much more recently. And this is an eastern mole, but it could be any kind of mole. This is a microscopic view of the whole head, cut at about this level here. And you can see the tiny little eyes that are, in fact, you can't see eyes here because the skin has covered over the eyes. And if you look in a little more detail, many of the ocular structures are still there, but they're disorganized. And again, you can imagine if this animal survived some catastrophic event and land animals all disappeared, and this animal came out again, well, probably the vision would return in this animal, but the nature of the anatomy would be different. Next. This just illustrates that this is estimate of visual acuity. And although primates among mammals have visual acuity that rivals some of the highly visible birds, a generic mammal like a cat here has visual acuity that equivalent to only the nocturnal birds. Next. So this is just to say that there's one other animal for which, one other type of animal for which this nocturnal bottleneck has hypotheses to come into play, and that's the snakes. Again, people think that snakes avoided dinosaur predation by going underground. And then next and last, and these are some features that reptiles have that snakes have lost: cartilage in the sclera, bone in the sclera, annular lens pad. They still have skeletal muscle in their eye. Photoreceptor oil droplets lost, and the shading of the photoreceptors also lost. So probably an evolutionary story with regard to vision, very much like what has happened in the mammals. And I think that's my last slide. Questions?
The end of the dinosaurs was marked by a catastrophic cosmic event. I forgot what it was called, but it's a black line.
Yeah, it was an well, there may have been other things as well, but most people believe that it was largely an asteroid that hit in the Yucatan Peninsula.
How does that fit in? Except
So,
at the end,
yeah. So it also ended the life of most mammals that had developed at that period of time. The animals that survived are the teeny dinosaurs, for instance, the birds, and also tiny mammals that were generalists that could feed on, that weren't very specialized in their lifestyle. And the same is true in other groups too. Tiny reptiles, which now have become lizards and et cetera, snakes. Those are those very small animals that are generalists in terms of their lifestyle are the ones that survive.
So their lifestyle of being subterranean basically protected them from the environmental changes that happened that killed off the dinosaurs.
I've never seen that. I've never seen that theory actually verbalized in the literature that I've looked at, but we were left to imagine that. Yeah. Yeah.
do current mammals and birds see color, and do they see it to the same degrees?
That's a great question. Current mammals, except for old world primates, which includes us, see in 3 colors, basically red, green, and blue. All other mammals see in 2 colors, which is basically blue and green. Most birds have not 2, not 3, but 4 color variant photoreceptors in their eye, and the additional one is in the ultraviolet, and that's true. A number of other even more primitive animals also have at least 3, sometimes 4 color variants. There are, in addition to that, there are other reasons to suspect that the experience of a bird seeing color is much more sophisticated than the experience of a mammal, but I'm not going to get into those. But birds almost certainly see color on a level beyond our imagination.
I've got a question about, to change the subject on a little bit, but insect eyes are completely different, I'm assuming. But how do they differ? And
Yeah.
Talk a little bit about when you first came in about different evolution in eyes. Different
Yeah, the eyes that insects use for visual acuity are called compound eyes. A compound eye evolves visual acuity by adding a repeating sequence of the same eye over and over and over again. So what they see is a pixel mosaic of from each one of those eyes. But it's a pretty complicated pixel mosaic because each one of those individual, we call it ommatidia, each one of those structures has 7 photoreceptive cells in it, including either 3 or 4 color variants, and then grayscale gradient detection as well. So each one of those pixels contains a lot of information.
Over here. What is moon blindness in a horse?
Moon blindness is an antiquated term for a condition we now call equine recurrent uveitis. And it's a very fascinating uveitis. It means an inflammation of the uvea. So it's a very fascinating inflammatory disease of the horse eye. One of the most common reasons why horse eyes are removed and sent to our lab. Has nothing to do with the moon.
Can it be treated?
People are learning more and more about treatment. Oftentimes with inflammatory conditions, the treatments are anti-inflammatory medication, which is the more and more they learn with surgical implants of locally drugs that can be released over time. They're more and more able to treat eyes with recurrent uveitis, but still it's one of the most common reasons why horse eyes are removed and sent to us. Yeah.
The difference of the animal's lifestyle, say a predator versus a grass eater, does that affect how the eyes develop?
Yeah. So most of what you're going to talk about in evolution and ocular anatomy is a rule of thumb. So it's not something that's carved in stone. But animals that are prey species, herbivores, etc., their goal in terms of vision is to have to be able to sense their whole environment. So they generally speaking have eyes on the side of their head, which gives them a very wide field of view, and they lack a certain structure in the retina that allows for both eyes to function together. Generally speaking, animals that are predators want to be able to zero in on something and then either pounce on it or chase it or whatever. They tend to have eyes that are in the front of their head so that both eyes look in the same direction. And then in the retinas, they'll have an area where if they're looking directly at something where both eyes will be looking together, it helps them with depth perception.
One more, Jim. Do raptors are like eagles, do they have some kind of a telescopic feature in the eyes that they could see from so far?
The telescopic feature of the eye is a high degree of visual acuity. It's like if you download an image from the internet that has 1,000 pixels versus one that has 200 pixels, you can magnify that image much greater. And they have a number of different features that allow them to have higher degree of visual acuity.
What's that? And what you see? Thank you very much.
Thank you.
Uh-oh, I got a red light. Is this working? Kind of. Gonna have to change the batteries here soon. Paula has our moment of reflection today.
Kevin.
Oh,
And
got it.
speak into this.
Oh, both?
Both of
Okay,
them, yeah.
got it. All right, with it being that time of year where many of us are out enjoying our gardens, including myself, I thought I would share a few garden quotes that inspire kindness or peace. Flowers are restful to look at. They have neither emotions nor conflicts, by Sigmund Freud. A society grows great when old men plant trees whose shade they know they shall never sit in. Greek proverb. I want it said of me by those who knew me best that I always plucked a thistle and planted a flower where I thought a flower should grow. Abraham Lincoln. And last, the glory of gardening, hands in the dirt, head in the sun, heart with nature. To nurture a garden is to feed not just the body, but the soul. Alfred Austin.
Do it this way. Okay, let me bring out the agenda. We have a couple of business items to take care of, which is namely president-elect, not co-presidents. We'll have to work out the co-presidency later on, but we have a nominee in Dick Hammerstrom for president-elect. Are there any others? And reading our bylaws and things, it's a very loose election process. All we ask is that the nominating committee deliver a slate, and how we vote, I couldn't really discern. So Dick, congratulations, president-elect. Let's see. No, some groups do that, but our bylaws don't say that's required. We could have had a second nominee last week, but 2 weeks ago I read the nominees and that's required.
Why don't we just nominate him 3 times today?
Because we don't need to do that. I don't want to do things we don't need to do. I want to keep things simple. So my understanding also is treasurer is appointed, correct? No, elected also. So Dick Hammerstrom has agreed to another year as treasurer. And then we'll figure out what happens when he's co-president, treasurer, well, exhausted, yes. And Richard is going to be secretary for another year. Thank you very much. And then we have 2 nominees for the board, including Leonard Massey, who's going to serve another year. And Jerry Schwach has agreed to step up for a 2-year term. So as far as my understanding, they are now elected. So congratulations. And Richard takes the next steps and sends them into international as our slate of officers for the year after next year, starting October 1st. Any questions on the election? And Meg agreed to help Dick with that second year, and we'll go from there. And speaking of presidents, Eileen is having some issues, physical issues, post-surgery. I'm not sure of the specifics at all, but you may want to send her a card, get well card maybe. So she could not make it today. Mike Ely has COVID, still negative with your spouse, but she has a fever. So I made the point about masks. I'm just more comfortable with them. I really don't want to get COVID again. Apparently the president survived quite well, but being double boosted, that's a big step forward, and I am too. So let's see, what else do we have? The board meeting is this afternoon, right at 1 o'clock. And what we're going to do with the board is to introduce a process for committee budget review. We have almost all of the budgets in. So we'll be just talking about the process that will vote on the budget next month at our board meeting. And Dick will explain some of that. Let's see. Announcements. Jerry, you have some. So if you do this, it works well.
All right.
Because they have to hear at home.
Got it. We had a blood drive last week at the Department of Revenue. And I'd like to thank Dick Hammerstrom, Tom Broad, let's see, John Esser, Bob Rand, and Co Williams, along with myself. There were 44 donors that showed up. Now, that's not a big number. We used to get in the 70s. But Department of Revenue people, I overheard somebody say they're only about 45% present in the building right now. We have another blood drive at Department of Revenue on September 29th. So that's a little bit over 2 months from now. Also, I received an email last week from the Department of Transportation asking us about when we were going to do our trash pickup on Highway 19. And she said, if we don't do it, they might take it away from us. And I know they aren't going to do it. And she told me that too. But they've got more paperwork for us to fill out in the future. And I told them, I said, you know, there's a bracket for one of those signs that's been missing since they put that roundabout in. And so we've only been able to put up one of those signs. And I told her, I said, that highway is a lot more dangerous now than it was when we started that about 18 years ago. So I think, I hope that they get the DOT out there to put that bracket up so we can put our sign up again. That's the least they can do for our safety.
So you're planning a fall cleanup, right?
Yes, we are.
Okay, any other announcements? Dick?
Whoop. Got my glasses there. Okay.
We need a little comedy routine here. Yeah, so we served at Wilmar on Saturday, and I also want to thank the people who helped out with that. Tom Broad, Ken Cossack, Co Williams, who isn't here, and Bob Rand helped out too. So we had, we only served about 27, 28 meals. It's just not, you know, COVID really knocked so many things apart, and we're now don't have the numbers back. As you recall, we used to serve at least 70, 75 people and over 100 when we did seconds. And so it was down. We served almost 40 meals, but again, people had seconds. Some people took home and everything. In any case, we served spaghetti and meat sauce and some vegetarian sauce in case Kevin showed up being homeless. And we had veggies and mixed fruit and Texas toast, and we had cookies. And of course, we had some of Co's homemade oatmeal chocolate chip cookies available. So.
Thank you. Okay, just any other announcements? I'll just go through some scheduling things. Next week, our speaker is, it should be an interesting one. The title is Madison's Mafia. It's by an author who apparently has researched this and has written a book on the underground world of Madison, probably. Can't say I've heard of it, but. Okay, he won't be here live, but zoomed in, which we're. That's right. We might have some members of that cult here. August 8th, Matt Rothschild will talk about 12 ways to save democracy in Wisconsin. And then we're still looking for somebody on the 15th. My backup plan as president is there's always issues to talk about with the club. Should we not be able to have a speaker or if the speaker doesn't show up. So as I said, the budgets of all except one committee have been submitted and the board will talk about those today. Last reminder is on the 29th of August, again, Oakwood Village has agreed to host our meeting. I talked with Jim. He's okay with that, obviously. And I did tell Jim, and we'll learn from Oakwood whether they are going to reimpose any kind of restrictions due to the significant rise in COVID. So be prepared to maybe come here on the 29th if they lock us out of Oakwood Village. But when do we have bingo?
It's a week from this Thursday.
So on the
31st is a Sunday. So the first-
1, 2, 3. So on the 4th, August 4th. So it'll still be well over 3 weeks in advance of that. So we'll see what happens, but they do have a mask requirement.
Right, we go in for bingo everywhere.
Yeah. And I may or may not, I'll talk to you about that if I can make it. But I think you owe me since I handled last time all by myself. Yeah, I want that star then. Put it on my notebook. Last announcement really is that we have only 2 more weeks of Miracle League. So the first week in August is the last game, which is 2nd, 3rd, and 4th. So if you haven't come out to see games, this week might be a really good one because it's not going to be warm and it's just going to be beautiful. Apparently on Tuesday, Wednesday, Thursday. So anything else for the good of the organization? I think we had 16 members I counted and our speaker and his wife. So thank you very much, Dick, for being a biologist. That was enthralling to me. I love evolutionary biology. And yes, the eyes are incredibly remarkable. So thank you very much for sharing, and we will see you next week, hopefully. I will call it a meeting. I get to do this.